Here's the pathophysiology of celiac disease. This is a really good image because it is going to basically summarize all the things we need to know about how the celiac disease occurs. Now, when we consume gluten, gluten has a protein called gliadin. Gliadin is ultimately going to be the protein that triggers this autoimmune destruction of our intestinal villi. And these intestinal villi are extremely important for the absorption of nutrients. Now, once gliadin enters our cells, our intestinal cells, it is going to be deaminated by tissue transfer glutaminase, which is found over here. Now, once gliadin is deaminated, antigen presenting cells are going to take up the deaminated gliadin. Now, the way you want to think about this is here's our antigen presenting cell, and our antigen presenting cells, they have a receptor outside their cell membrane called MHC2 receptors. And these receptors, they're basically going to take up antigens and present it to T helper cells. Now, the antigen that it is going to show to our T helper cells is going to be the deaminated gliadin. Now, these MHC2 receptors, they're basically proteins that show antigens to our T helper cells. These are encoded by genes called HLA genes, alright? And these HLA genes are for people with celiac disease. The HLA genes that code this receptor are going to be HLA-DQ2 and HLA-DQ8, as we see over here. Now, once antigen presenting cells present the antigen or the deaminated gliadin to our T helper cells, there's going to be two things that are going to occur. The first thing is going to be the stimulation of plasma cells to produce antibodies. So we have the antibody against the tissue transfer glutaminase, we have the anti-endomysial antibody, as well as the anti-deaminated gliadin IgA antibodies. These are going to be found in the blood work of a patient with celiac disease, and they can be used as markers to diagnose a person or a patient with celiac disease. Now, the second thing that is going to occur is our T helper cells, they are also going to produce cytokines. And the cytokines are basically messages, and they're going to signal the fibroblasts to produce proteases, and these proteases are going to destroy the villi that are found in the membrane of our small intestinal cells. And this is going to result in malabsorption of nutrients. This is called intraepithelial lymphocytosis. It's basically the finding of lymphocytes within our brush border. And as a result, it is going to ultimately result in the loss of the brush border